excited to be able to share yet another conversation with Dr. John Cannon from McMaster University. In this discussion, we're going to talk specifically about my experiences.
Yeah, how are you doing?
I'm doing well. So we talked in July. It's been quite a journey with the start of the semester,
Hmm.
and I am more and more um, convinced and empowered by your last email, convinced that my perception of time and rhythm and my manifestation of time and rhythm as being separate entities, when I find myself having issues, I'm finding myself more and more beginning to tap into my perception, almost as if I'm getting myself out of myself and
Yeah.
observing myself in the third person, if that makes any sense at
Yeah.
all.
So I mean, I think you're learning to do intentionally what a lot of people with Parkinson's learn to do just implicitly. Because there's a lot of evidence that people are using cerebellar circuits to compensate for basal ganglia circuits with Parkinson's. There are thoughts about how rhythmic auditory stimulation helps people walk that are all based on the idea that they're recruiting the cerebellum to do motor and potentially timing things, but motor things mainly that the basal ganglia would otherwise be doing. What I sent you is a sort of circumstantial but interesting bit of evidence that the amount of matter in your cerebellum predicts how good you are at figuring out whether a tempo is faster or slower than another tempo.
What you're telling me is that I'm not crazy, that you believe that the part of my brain that is perceiving rhythm and the part of my brain that is in charge of manifesting rhythm are different places.
Uh, I would not say I'm quite certain about that. I would say that I've been chasing down that theory, or at least some version of that for some years. So one of the experiments going on in my lab is a an eye blink conditioning experiment, getting a cue and then learning that you need to blink a certain amount of time after that cue. And you can get conditioned to blink 500 milliseconds later, 600 milliseconds later, or something like that. And in animals, at least, that is like entirely happening within the cerebellum. And in humans, we can sort of do it cortically as well. There's like other things that are participating, but there's this cerebellar timing component. Um, so it's very much involved in timing intervals. Um, and people just don't know very well how involved it is in making predictions and parsing a rhythm. There's a guy in Denmark, Lars, what's his name? Okay, hold on, Anderson, Lau. That's it. Lau Anderson is doing a series of MEG experiments with people with Parkinson's and healthy individuals, and is looking at periodic tactile stimulation and trying to see how the basal ganglia and the cerebellum are interacting as we do things with that. And I think he's got strong evidence that at least in the sort of slower periodic tactile stimulation, the cerebellum is really important for keeping time. Then there's this group in Japan, the Tanaka lab, that have been doing neurophysiology in primates. And there is one particular paper that I'm happy to drop in the chat here. I'll just read you the abstract. It's really helpful. Both the cerebellum and the basal ganglia are involved in rhythm processing, but their specific roles remain unclear. During rhythm perception, these areas may be processing purely sensory information, or they may be involved in motor preparation, as periodic stimuli often induce synchronized movements. Previous studies have shown that neurons in the cerebellar dentate nucleus and the caudate nucleus exhibit periodic activity when the animals prepare to respond to random omission of regularly repeated visual stimuli. So they're using an omission paradigm where they leave out one thing in the rhythm, and they see what parts of your brain are responding, or what parts of the monkey's brain are responding. To detect stimulus omissions, the animals need to learn the stimulus tempo and predict the timing of the next stimulus. The present study demonstrates that neuronal activity in the cerebellum is modulated by the location of the repeated stimulus, and that the striatum by the direction of the planned movement. However, in both brain regions, neuronal activity during movement and the effect of electrical stimulation immediately before stimulus emission were largely dependent on the direction of movement. So there was a spatial component here that doesn't line up very well with the sort of timing, auditory timing. But here it says the results suggest that during rhythm processing, the cerebellum is involved in multiple stages from sensory prediction to motor control, while the striatum consistently plays a role in motor preparation. Thus, internalized rhythms without movement are maintained by periodic neuronal activity, with the cerebellum and striatum preferring sensory and motor representations, respectively.
So what is that in English now?
In English, okay, focusing on the last sentence, the internalized rhythms without movement are maintained as periodic neuronal activity. There's periodic things happening in your brain that line up with the rhythms, with the cerebellum preferring sensory representations, meaning the cerebellum is more involved in just sensing the rhythm and making internal predictions that have nothing to do with your physical movement. Whereas the striatum prefers motor.
Where's the striatum? What is the striatum?
Oh, I'm sorry. Right, the striatum is the area that's impaired in Parkinson's disease. I mean, there's multiple areas, but one of the most severely affected areas is lack of dopaminergic neuromodulation in the striatum. So it's the input nucleus for the basal ganglia. Okay. Sorry. Then to backpedal a little bit. The basal ganglia prefers motor representations of rhythm. The cerebellum prefers sensory representations of rhythm. So that I think is what you're looking for.
Boom.
Right?
That's hello.
Yes.
That's me.
Yes.
So now when somebody does that, when somebody does a document like that, I mean, at what point does that become an accepted point of view?
Good question. Yeah. So that's as of 2023. That's when that experiment was done. It is still, people are still talking about it a lot. I mean, people in my tiny universe are talking about it a lot. And
I like your universe because you're talking about my world.
Yeah. No, I can tell. And I'm really interested in your world because it's a living world for the dry academic conversations that we're having about these brain areas.
That's fascinating.
So, I mean, I think, there's also, I think that paper, I think, is sort of the smoking gun. I think because like it's extremely invasive and direct recordings from these brain areas.
I have Parkinson's on my right side, and of course that's the side that I have I hold the baton and I manifest tempo.
Yeah.
And so for me, I perceive it. I perceive my good perception of rhythm on my left side because because that's whether or not it's there.
Yeah.
And so I perceive like when I'm having an issue, I almost just like lean more on my left side. But I'm telling you, man, it's making a difference.
Yeah, but like what those areas are doing. I mean, the simplest way to talk about what might be happening is to say that there is a rhythmic timekeeping process, which is to say neural activity that aligns itself to every beat that is happening in a loop between motor areas and the basal ganglia, and that requires the basal ganglia to be actively sending signals to these motor areas, possibly just to control the tempo. It's possible that you identify a tempo and then basal ganglia sets the tempo for your motor areas to keep time. But that at the same time, the cerebellum is when it gets a beat related cue from either what you're listening to or internally, it starts its own timing process in the granule cell layer, where this layer of neurons, they're all talking to each other and can make elaborate, long-lasting patterns of activity. And then when the next click comes in, so the cerebellum has learned that at a particular moment, now that you've listened to the beat for a few cycles, it needs to predict that there's going to be an event at this particular time, at this particular place in the granule cell's pattern. And if that prediction is early or late, you're able to detect that by doing some kind of comparison. I don't know exactly what, but like basically, this is a really long way of saying that one way of talking about this is that there are just two different timing processes happening in two different parts of your brain, each of which can align to the beat, and one of which is inherently motor, is inherently um, keyed into everything you're doing with your body, and your body can sort of lead it. If you start moving your body, then you start picking up the rhythm with this area of the brain, and if you're inclined to move to it, that means you're picking up this rhythm with this area of the brain, with this loop between basal ganglia and supplementary motor area, this motor area of cortex. And then at the other side, there's this other timing process in the cerebellum that is listening to the specific intervals, and quickly learns that, oh, since I've experienced 3 or 4 intervals of 600 milliseconds from one beat to the next time I need to predict that 600 milliseconds later there's going to be a beat. And it also does some long-term learning and is able to say, like, okay, if there's a beat at this time, then we learn this cognitive representation of it. So it's done a lot of learning over the course of listening to a lot of different tempos, and is now able to just recognize that interval, that amount of time between beats. And so it's much less like part of our regular sense of rhythm, embodied sense of rhythm, and it's more about this absolute interval timing. And that's one whole perspective on this. And I'm kind of skeptical of that a little bit. I guess I think that the cerebellum may be also listening to. I don't know. I don't really know what's happening, but I'm trying to explore it. I'm trying to make models to articulate in this rigorous way what I think these areas might be doing, and articulate these different hypotheses as concretely as I can. And then I'm trying to find these like little low lift experiments that can really start to help disambiguate. So that's a long way of saying, I guess, that we can point at brain areas, and people are pointing at the cerebellum and saying, like, this is involved in rhythm, but it's involved in some kind of sensory representation of rhythm. It's involved in tempo discrimination, but we don't know why. Yeah, I've been blabbering. I can actually. I'm interested to just hear more about your experience of this. Like, how did you sort of figure out that you were able to do this?
Well, I mean, because of my condition, I'm not as active conducting as I used to be. I used to, be especially in rehearsal, conducting everything, controlling everything visually, and I just simply can't.
Yeah,
And so a lot of time in rehearsal, when things are grooving and everybody's playing together, they're not watching me for a beat anyway.
yeah,
And so I will often now in rehearsal kind of just stop conducting and monitor.
uh-huh.
And as soon as I stop conducting and start monitoring, I actually uh, hear a lot. I hear a lot better
Yeah, yeah.
in
So
terms of,
actually stopping moving becomes part of this
well,
equation.
without a doubt. In many regards, my perception of rhythm is heightened now because of my condition. I think I'm more sensitive now to changing rhythm than I was ever before this Parkinson's journey began. And but for me, that is a completely different thing than my ability to move and my ability to have confidence in demonstrating and initiating tempo visually, physically.
Yeah. So here's something you might think about. A puzzle for me from the very beginning of my involvement in rhythm science has been that on the one hand, it seems like when we go to a faster tempo, we speed up a clock in our heads. We speed up some process, and when we go to a slower tempo, we slow down this process. But if the clock is changing speeds, then we don't have a clear way of saying this is running faster, this is running slower, right? We need a separate clock that's not changing speeds to tell us whether something is going faster or slower.
It's the relativeness.
Exactly, it's the relativeness.
So that there are in most pieces some kind of temporal relationship when a changing tempo. Very seldom is there a changing tempo that there's not some sort of relation. So I don't it for me I would not characterize it as having a separate clock. I would say that the clocks overlap. And
Yeah.
before I jump to the new clock, I'm perceiving the what that new tempo clock will be while I'm finishing up the initial tempo. They overlap.
Yeah, so I mean that's a puzzle to me because like that skill is one that is I mean I think it's really it runs contrary to the way that we usually use our brains. Like people experience one beat. You know, you even when you're listening to a polyrhythm, it doesn't have two beats, it has one beat. Parts of what we do are dependent on absolute timing, and parts of what we do scale with relative timing. And though this Tanaka Lab 2023 paper is sort of the first one to really do this dissociation between motor and sensory representations of rhythm, a lot of people over the last 15 years have pointed out that in fMRI, the basal ganglia shows up for relative timing tasks, and cerebellum shows up for absolute timing tasks. A number of fMRI studies, the magnetic resonance imaging, where you get really good spatial resolution, but no time resolution, or limited temporal resolution. A number of those studies have shown that when you give somebody a task to do that requires them to make judgments based on the absolute timing between two cues, then they seem to be drawing more heavily on their cerebellum. And when you give them a task to do that involves saying whether something is, you know, continuing a steady rhythm, but regardless of the actual exact interval, regardless of tempo, something that's relatively timed, then that the basal ganglia lights up more. And so I mean I think that is a body of literature where if you treat tempo judgment as an absolute timing task, which it kind of is, you know, you need to have that clock that's not speeding up or slowing down. You need to be able to judge it relative to some fixed clock. Then it kind of it makes sense that you would be doing that with the cerebellum, which is an area that is not at all impaired in Parkinson's.